Hello guys, and welcome back all again to another video tutorial by Genome Biotech. In this particular video tutorial, we would be talking about the genome complexity as we have already learned through PowerPoint animations as well as videos. Now we are going to talk about the satellite DNA. In my previous lecture, I have explained to you through animations that how the satellite DNA moves from one location within the genome to another location. Now what we have to study now in this particular lecture is that in what manner the insertional sequences or the bacterial is sequences differs from any other DNA transposable elements. We know that the transposable element can be of two different types. One, it could be a simple transposable element, or second could be the complex transposable elements. Okay, now we will have to categorize which is the simple and which is the complex transposable element and how. Now you look here, insertional sequences are the most common transposable elements that are going to be present in the case of bacterial cell. Okay, so this insertional genomic sequences is elements are going to be the transposable element that are going to be present in bacteria. The journal structure of the is element, when you are going to look, it is being described in the diagram below. In this particular diagram, you can find that the transposable element has a protein coding gene. This is the protein coding gene, and in the animation we have already discussed that the protein coding part of this is element is actually the one that is going to encode the transposase enzyme. Transposase enzyme is going to encode the protein coding part. And so here we are going to write about the enzyme, and the enzyme is referred to as the transposase. Okay, so which is the enzyme which is being involved? The enzyme that is being involved is transposase. This is transposase. We always know that if the genetic mobile genetic element or the transposon just encode for transposase, then it is referred to as simple mobile genetic element. Simple mobile genetic element, when it is encoding only for a single transposase, then it is called as simple mobile genetic element. But when you talk about a composite mobile genetic element, the composite mobile genetic element will encode not only transposase, but another gene along with the transposase will be present, such as an antibiotic resistant gene or any other gene. Okay, so then it will be forming the structure of the composite transposable element. Okay, just reading line by line. So is element is diagrammatically depicted below. It consists of an inverted repeat sequences which are about 50 base pairs. Invariably is present at each end of the insertional sequence. These repeats are called as short direct repeats. These are the inverted repeats, and then there is the direct repeat. The presence of direct repeat is a hallmark of all the different transposons. दुनिया में जितने भी ट्रांसपोजोन्स होते हैं उनके लिए the presence of short direct repeat is actually a hallmark that indicates that yes, a short direct repeat is present towards the end of the chromosome. These are the short direct repeat, and then there are inverted repeats which are present. The inverted repeats which are present, once the inverted repeats are present, the inverted repeats are the one that are being created due to transposition. ट्रांसपोजिशन की वजह से इन्वर्टेड रिपीट्स क्रिएट होती हैं. Please, I just request you all to go through the previous lecture, which Shivani you have already missed. I am uploading that particular lecture. You just go through that particular lecture. In that lecture, you will get all the animations of this particular topic. I have already explained this topic with animations, and this is a theoretical class. Tomorrow also, our class would be of animation first, and then it would be a theoretical class. So in molecular biology, everything will be shown to you via animations, and then a class would be taken. Okay, so this is how the simple transposable elements occurs, where whereas the composite transposable element will encode along with the transposase another gene. Okay, another gene will be present. Mostly the antibiotic resistant gene will be there. The enzyme that catalyzes the transposition of the is element from one location to the other is referred to as the transposase enzyme. Just to give you a view of how does the transposase enzyme work and in what manner the transposase enzyme is going to move the DNA from one location to another, we are also going to look at the animation point. Okay, give me a second. Just starting the animation in front of you for you all. Here we are. Just look here. See, this is how
Transposition is a form of genetic recombination that moves defined genetic elements, called transposable elements or transposons, from one DNA site to another.
Although we have already finished this, you can just go
When
ahead
you
and
have
revise
completed
it
this
from
exercise,
the
you should
previous lecture.
understand the cut and paste mechanism of transposition and understand the replication.
Just a second. Some problem. Okay, see,
Transposition is a form of
this is transposition. We have already dealt with this.
the recombinases responsible for transposition are usually
This is the enzyme that is responsible for transposition. This enzyme is referred to as transposase. Now, how does transposase is being encoded? Transposase is being encoded by the gene, which is being encoded within the transposons. Okay,
usually called transposases. Transposons carry a gene encoding their own transposase.
Now, this is what I'm telling you. The transposons, which always include a gene that is referred to as the transposase gene. And here is the transposase gene which is present. Now, what is this transposase? Transposase is a protein that has the ability to act as an endonuclease. Okay, it is not a restriction endonuclease, but it is an endonuclease. It can cleave the DNA by breaking the phosphodiesterase linkages. So yes, in our body also there are places where transposons, where endonucleases are present. These it serve as an endonuclease. As it serve as an endonuclease, so it has an ability to cleave the DNA at specific locations. Okay, and then to move the transposons genes from one location to another, then it is referred to as cut and paste mechanism of transposition. See what is going to happen. See how this process of transposition is going to take place.
Transposons may carry a few additional genes as well.
Okay.
For example,
this is what I was telling you. When it contains only a transposase gene, then it is referred to as a simple transposons. Now, when along with the transposase gene, there are other genes which are also present, then it is referred to as a composite transposons. So the simple transposons and the composite transposons are going to be different from each other. Okay. Video is not visible. Hello, is the video not visible? Okay, now it is now visible now.
The recombinases responsible for transposition are usually called transposases. Transposons carry a gene encoding their own transposase. Transposons may carry a few additional genes as well. For example, many bacterial transposons carry genes for antibiotic resistance.
Okay, so this is a type of transposons which are referred to as the simple transposons. Simple transposons are the one that contains the gene for themselves only. So when they contain a gene only for themselves, then they are said to be the simple transposons. Simple transposons. Okay. When along with the transposons, when along with the simple transposons, other genes are also present, then it is referred to as composite transposons. Then it is referred to as composite transposons. Such type of transposons are called as composite transposons. Okay, moving forward. Next point.
The very ends of a transposon's DNA are arranged as inverted repeats. These inverted repeats range from about 25 to a few hundred base pairs and carry the recognition sequences for transposase binding.
Okay, So these transposase binds to inverted repeat sequences, and these are the palindromic repeat sequences. As they are inverted repeats, so they can be recognized by the transposase enzyme. The transposase enzyme are going to recognize the inverted repeats. When the transposase enzyme are going to recognize the inverted repeat, they are going to cleave the inverted repeats. As the transposase enzyme cleaves the inverted repeats will be able to move from one location to the other, and the transposase itself find its target location in order to transpose the transposable element from one location to another. Transposones enzyme have the ability to transpose the genetic material from one location to another. ये जो transposase enzyme है यही recognize कर लेता है inverted repeats को, और inverted repeats को recognize करके उन्हें cut करके इस transposons को एक जगह से दूसरी जगह ले जाता है.
Simply put, cut and paste transposition involves the excision of a transposon from its initial location in the genome, followed by integration of this excised transposon into a new DNA site. This is a non-replicative mechanism, meaning that the transposon is not copied during the transfer.
Okay, in this method the transposon is not being copied, so it is not being copied from one location to the other. It has not been copied. That means the number of copies of this transposon will remain the same, so it has a number of copy that is equivalent to 1. Next.
We will now look at the cut and paste mechanism in more detail. To initiate transposition, transposase subunits bind to the terminal inverted repeats.
Okay, these are the terminal inverted repeat to which the transposase subunits are going to bind. As the terminal inverted repeats are the site where transposase subunits are going to bind, they will cleave the DNA molecule here. How?
Once the transposase subunits recognize these sequences, they bring the two ends together to generate a stable protein DNA complex called a transpososome or synaptic complex.
Now,
This complex
it leads to the formation of a complex that is referred to as transpososome, or also referred to as the synaptic complex. It is this complex where the cleavage of the DNA is going to take place. Next point.
functions to ensure that the DNA cleavage and joining reactions occur simultaneously on both ends of the transposon, and also protects the DNA ends from cellular enzymes during transposition.
So here, the ends of the DNA are being protected from the cellular enzyme so that the cellular enzyme should not degrade the ends of the DNA molecule. And these ends of the DNA molecule are now being effectively protected from the cellular enzymes. Next point.
Next, the transposon is excised from its initial location in the genome. To accomplish this, each of the transposase subunits nicks one DNA strand, such that the transposon sequence terminates with free 3' hydroxyl groups at each end.
Now there is a termination that is going to take place, and the free 3' hydroxyl group is going to be produced. There are two free 3' hydroxyl groups which are being produced. One is the 3' hydroxyl group which is being produced here. Another is the 3' hydroxyl group which is being produced here. And now the excision is going to take place.
To finish the excision reaction, the other DNA strand at each end of the transposon must also be cleaved. Different transposons use different mechanisms to cleave these second DNA strands. For some transposons, an enzyme other than transposase directly cleaves these strands.
Okay, there are two different procedures. Either the different enzymes can be used to cleave the same strand, or the same strand can be cleaved alternatively by the transposase made free 3' OH. This 3' OH may cleave the other strand, and this 3' OH may cleave the complementary strand. So it depends upon how the cleavage reaction is going to take place. Next point.
For other transposons, the transposase catalyzes attack of the uncut ends of the DNA strands by the 3' hydroxyls directly opposite them on the other strand. This reaction forms DNA hairpin intermediates. The two hairpin ends are subsequently hydrolyzed by the transposase. Some transposons use still other, more complex mechanisms to cleave the other DNA strands.
Now what is going to happen? Sorry for the disturbance caused. See, These can be alternatively broken down, and here there is a double stranded DNA nick that has been created. There is the double stranded DNA which has been broken down, and as the double stranded DNA has been broken down, so this transposon is going to move from one location to another. As you find that there is a double stranded break which has been produced, if this double stranded break is produced in the case of prokaryotes, then SOS repair system will operate. Which repair system will operate? SOS repair system will operate to heal the gap. Okay, healing of gap, healing of gap produce, healing of DNA break. DNA break is going to take place through SOS repair system. SOS repair system, which will involve certain proteins. Can you name all the proteins? Rudra, try to name all the proteins. Okay, the first proteins are Y family DNA polymerases. Y family DNA polymerases refers to template independent DNA polymerases. I have already told you all. These include DNA Pol 4 and DNA Pol 5. DNA Pol 4 as well as the DNA Pol 5. Along with the DNA Pol 4 and DNA Pol 5, these DNA polymerases are also referred to as UMUD2 DNA polymerases. UMUD2. UMUD2 DNA polymerases. DNA polymerases, which are going to polymerize the DNA molecule and repair the cleaved strands. Okay. There would be another protein, Ku protein, Q protein, that is going to take part. So these proteins will be involved in the repair process. देखिए क्योंकि डबल स्ट्रैंडेड डीएनए यहाँ पर ब्रेक हो चुका है और डबल स्ट्रैंडेड डीएनए को आपको रिपेयर करना है, तो डबल स्ट्रैंडेड डीएनए को रिपेयर करने के लिए आपके पास में एक प्रोटीन का एक सिस्टम होता है. किसके अंदर? ये सिस्टम केवल और केवल प्रोकैरियोट्स के अंदर है, बैक्टीरिया के अंदर है. इट इज प्रेजेंट ओनली इन प्रोकैरियोट्स. इट इन्वॉल्व्स अ प्रोटीन कॉल्ड एज वाई फैमिली डीएनए पॉलीमरेजेस, also called as DNA polymerase 4, DNA polymerase 5, UMUD2 DNA polymerase, and a protein that is referred to as Q protein. Okay, so these are the proteins that are going to be involved. Next point. Now, deleting this out, you'll get the recorded version. You can also look at the recorded version, or you can take snapshots at different time interval. It is also allowed. Okay, next point.
The 3' hydroxyls of the transposon DNA then attack the phosphodiester bonds at the target DNA. This reaction is called DNA strand transfer. The sites of attack on the two target strands are usually separated by 2 to 9 nucleotides. This distance is fixed for each type of transposon.
Okay, this distance is going to be fixed for each type of a transposon molecule, and this transposon will now be inserted into the target site. ये transposon अब अपनी target site के ऊपर insert हो जाएगा. This target site is actually being recognized by the transposase enzyme, which recognizes a particular site, and to that particular site will transposase the double-stranded DNA molecule. It will transpose the double-stranded DNA molecule to a particular site.
The gap in the target DNA resulting from the staggered attack on the two strands is then filled in by cellular DNA repair proteins. The 3' hydroxyls of the target strands serve as primers for DNA replication. Filling in these gaps gives rise to DNA duplications that flank the transposons. These sequences are called target site duplications.
Now see this. There are certain target sites, and here the inverted repeat sequences have been duplicated because these inverted repeat sequences are being produced as there have been an action of DNA polymerase enzyme. So this phenomena is called as target site duplication mechanism. इस phenomena के अंदर target site का duplication हो गया है क्योंकि DNA polymerase ने अपने complementary DNA को replicate कर दिया है. Next.
The double-stranded breaks at the initial site of the transposon must also be repaired. These breaks can be repaired by homologous recombination or joined directly by the non-homologous end joining pathway.
I have already told you that in the case of prokaryotes, when this gap is going to be produced in the case of prokaryote, there would be an SOS response, whereas in the case of eukaryote, there would be homologous end joining that is going to take place. Next point.
Way. Some transposons move by a replicative mechanism.
Now, see, the movement of the transposons can take place by two different mechanisms. One is referred to as the conservative mechanism and another one is referred to as the replicative mechanism. Now, there can be two different types of mechanism of transposition. One is referred to as the conservative mechanism. I have already told you conservative mechanism. Another one is called as the replicative mechanism. Now, what happens in the case of conservative? What happens in the case of replicative mechanism? In the case of conservative mechanism, the copy number of the transposon will remain the same. अगर आप बात करते हैं conservative mechanism की, if you talk about conservative mechanism, the copy number of the transposon remains same. Copy number of the transposon remains same. Of the transposon remains same. Then it is referred to as conservative mechanism. When the copy number of the transposon is going to be increased, then it is referred to as replicative mechanism. When the copy number of the transposon, when the copy number of the transposon is going to increase, it is called as replicative mechanism. Moving forward.
Although the outcome is different, the mechanism of strand transfer in replicative transposition is very similar to that of cut and paste transposition.
See this
However, the transposon DNA is duplicated during transposition, leading to two copies of the transposon joined to the new and old DNA target sites.
Now, this is what we have seen. In the case of replicative transposition, the DNA has moved from one location to another, so that now the copy number of the DNA has been increased. First, there was a single copy of a DNA molecule. The same DNA has been now present in two copy number. Then it is referred to as replicative transposition. In the other cut and paste transposition, what has happened is that the DNA has been made to cut and this DNA is going to move from one location to the other. Next. Here, in this particular case, even recombination can take place. As the recombination can take place, then it will produce new type of combinations that are going to occur. Okay, next. Next point.
Just as in...
Now, I'm just deleting all these things because they might be producing trouble to you. Okay.
In cut and paste transposition, replicative transposition begins with the assembly of a transpososome.
Now, this is the complex that is being produced. That is referred to as transpososome. And this complex will mediate both the cut and paste mechanism as well as the copy and paste mechanism. And now how the cut and paste and copy and paste mechanism are going to operate. Please look.
Next, the transposase subunits introduce nicks in the 3' ends of the transposon strands. However, in contrast to cut and paste transposition, the transposon DNA is not excised from its initial location.
Okay. Next, the transposases are not being excised from their initial location. Next point.
As in cut and paste transposition, the 3' hydroxyls of the transposon DNA then attack the phosphodiester bonds at the target DNA. However, the 5' ends of the transposon sequence remain joined to the initial flanking DNA. The replicative transposition intermediate therefore consists of a doubly branched DNA molecule.
There is a thus a replicative transposition intermediates is being produced, and this replicative transposon intermediate which has been produced, it consists of a doubly branched DNA molecule. So this is a double branched DNA molecule that has been produced. Now they would be separated. Moving forward.
The two DNA branches within this intermediate each have the structure of a replication fork. The DNA replication machinery can then assemble at one or both of the forks. The 3' hydroxyls of the nicked target DNA serve as primers for DNA synthesis. Replication proceeds through the transposon sequence, generating two copies of the transposon DNA. Once again, the staggered attack of the target DNA results in target site duplications flanking the element.
Now again, target site has been duplicated and there are two copies of the target site that has been produced. That means inverted repeats are now present in two copy number, and here inverted repeat is present. This transposase is only present in a single copy number. So that's the major difference between the two. Next.
In the process of moving, replicative transposition often rearranges the flanking chromosomal sequences. For some transpososomes, site-specific recombination occurs between the two transposons, resolving these rearrangements.
Okay, so site specific recombination is going to take place and new combinations are going to be produced. Next point.
How well do you
I have already finished this particular part, so I'm not going into it there detail. Now, moving back to the Pathfinder, what we were discussing about was molecular biology notes. Okay, so here we were talking about the IS element. These are referred to as the simple transposable element. In addition to IS element, bacteria may even contain sometimes the composite mobile genetic element. These composite mobile genetic elements are also referred to as the complex transposable element or complex mobile genetic element. These complex mobile genetic element include specific genes such as the chloramphenicol resistant genes as well as the tetracycline resistant genes. इनके अंदर कुछ specific genes होते हैं, and those genes which are present within them are the chloramphenicol resistant genes as well as the tetracycline resistant genes which are present. Okay, so there is the chloramphenicol resistant gene and the tetracycline resistant gene which is present here, and both the presence of both these genes indicate that we are going to talk about the composite mobile genetic element. Now, eukaryotic transposition. Now, what do you mean by eukaryotic transposition, and how does eukaryotic transposition takes place? The eukaryotic transposition is similar in structure to bacterial IS element. Now, eukaryotic transposons are much more similar in structure to the bacterial IS element, except that the eukaryotic transposons have certain other features. They may be activating transposons, or they can be associating transposons or dissociating elements. These were the two elements that were firstly being discovered by Barbara McClintock. The P elements of the Drosophila. The P elements in Drosophila is one of the best example of the transposable elements that occurs in the case of eukaryotes. This element is 2907 base pair long and features 31 base pair inverted repeat which is present towards their end. Now, this is the length of the transposable element, and these are the inverted repeats which are present. These inverted repeats will be the site where the restriction in the nucleus are going to act. P element do not utilize an RNA intermediate during transposition and can insert at many different positions in the case of Drosophila chromosome. Within the chromosome of the Drosophila, the P elements can be inserted at many locations within the genome. Next point. The other type of transposable elements are the autonomously controlling elements and non-autonomously controlling elements. Autonomously controlling elements are the elements which can move from one location to the other. Then they are called as autonomously controlling element. Autonomously elements are capable of transposition, okay, from one location to other, whereas non-autonomously transposable elements have a mutation within the transposase within the gene coding for transposase enzyme. In non-autonomously controlling elements में जिस जीन में ट्रांसपोजेज एंजाइम को इनकोड करना चाहिए उसमें म्यूटेशन हो गया है, there has been a mutation in the gene encoding for the transposase enzyme. A mutation has been associated. A mutation has been associated. A mutation. 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 Has been associ associated in the genes encoding for transposase, encoding for the transposase enzyme, for the transposase enzyme. Okay, as there has been a mutation, thus the enzymes will lose their ability. Thus you might think that the transposons will lose their ability from locomotion from one place to another, so the transposons will remain fixed, but it is not so. Now supposingly this is a double stranded DNA molecule, just making a double stranded DNA molecule. Okay, if I have made a double stranded DNA molecule that could be looked upon here. Now supposingly this is a double stranded DNA molecule, there is a 5 prime, 3 prime, and a 5 prime and a 3 prime end which is present. And there are two different sequences which are present. One is autonomously replicating sequences, and the red color indicate defective non-autonomously replicating sequences. These are non-autonomously replicating sequences. The other are autonomously replicating sequences. Okay, these are autonomously replicating sequences. So these autonomously replicating sequences will encode an enzyme called as transposase. It will encode an enzyme, and the enzyme that will be encoded is referred to as transposase. This transposase which is present, this transposase will go here and will also catalyze the transposition of another DNA molecule. ये यहाँ पर जाता है और non autonomously transposable elements का भी transposition करा देता है. It will cause the transposition of even the non autonomously transposable element. Transposase enzyme of a transposon will also catalyze the transposition of the transposition of a defective DNA molecule, defective transposon. Okay, so if there is a defective transposon, then that defective transposon, also referred to as the DS element, could also be made to move with the help of an element that is referred to as the AS element. Do you know who discovered these transposons? Transposons were firstly discovered in Zia maze by by Barbara McClintock. Barbara McClintock. And she was the one who discovered the AC element as well as the DC element. It was Barbara McClintock who discovered AC element as well as DS element. This is what her discovery came to. This came to the conclusion that the DS elements didn't have the ability to encode for transposase enzyme, but because of the AS element, they have the ability to move from one location to the other. Okay, now moving forward, is this clear? You can take a snapshot. I'm just going to delete this out and move forward, clearing all the drawings. Now moving forward, retrotransposons. Now, what are these retrotransposons? How retrotransposons are going to operate? What do you mean by retrotransposons? Retrotransposons are the one that are going to move from one location within the genome to the other location in the form of an mRNA transcript. Okay, so they would be moving in the form of an mRNA transcript, and then that mRNA will be getting converted into a DNA molecule. Or eukaryotes contain retrotransposon mobile DNA element that transpose as an RNA intermediate utilizing a reverse transcription mechanism. So they are going to move from one location to another through the action of reverse transcriptase enzyme. It is this reverse transcriptase enzyme that is going to catalyze its movement from one place to another. Okay, next. Now, there can be these mobile genetic elements are divided into two major categories. One is referred to as viral mobile genetic element, another one is referred to as non-viral retrotransposons. These mobile genetic elements are divided into two major categories. One is referred to as viral, another one is referred to as non-viral. Okay, first could be viral, another one could be non-viral. Now, viral retrotransposons. Viral retrotransposons are flanked by 250 to 600 base pair long terminal repeat sequences. Okay, when you talk about viral retrotransposons have long terminal repeat sequences, and these long terminal repeat sequences are made up of 250 to 600 and base pair long LTR regions. Okay, similar to those in retroviral DNA, and like retroviruses, always encode reverse transcriptase and integrase enzyme. So these are going to encode for reverse transcriptase as well as the integrase enzyme. They are going to encode for both of these things, reverse transcriptase as well as integrase. They move in the genome by being transcribed into RNA, which then undergo reverse transcription and integration into the host chromosome. So they are going to move in the form of an RNA intermediate through reverse transcription and integration into the host cell chromosome. Viral retrotransposons are abundant in yeast, and some of the viral retrotransposons that are present are the tie element and tie element in the case of yeast, and in the case of Drosophila, the copia element. So these are the two elements which are present. One is called as tie element, another one is referred to as copia element, which occurs in the case of Drosophila melanogaster. So these are the two different sequences that are being used. Another one is referred to as non-viral retrotransposons. So, what are these non-viral retrotransposons? How these non-viral retrotransposons moves from one location to another, that we are going to look forward here. These non-viral retrotransposons include the two most abundant non-viral retrotransposons that we have to study are the line, long interspersed nuclear element, and the sign, short interspersed nuclear element. So it's almost 10:15. I will conclude here only, but tomorrow we have our both. I will take both the classes, so that would be an elongated class. Plus, if you can please do arrange for the seventh edition of the Pathfinder, it would be much more better. I'm asking in the group, if someone has it, please do share. We are going to study this particular topic about the line and the sign sequences from the seventh edition of the Pathfinder. Okay, any doubt, any problem, if you have, you can ask me, and the recorded version of the lecture will also be shared in our group. In the meantime, that's all for today, because we have to study about the line and the sign from the next book, that is the seventh edition of the Pathfinder, because there are informations which are given there which are not present here. So we would be looking at that in tomorrow's lecture. Okay, that's all for today. If you have any doubt, you can always ask me in the group. Thank you. Good night. Bye bye.